Hey, so welcome back to Bio 100, and this is where we left off at our last lecture, showing this and wondering if you guys could make sense of any of these images and what was happening here as we kind of begin our discussion of genetic engineering. I'm not sure how many of you were even curious enough to look these up, but we'll kind of walk through each one of these carefully together. So what we have here is a cat. Cats, clearly a cat, sorry. But cats have actually their own form of HIV. And what scientists are able to do is go in and try and cure the cat form of HIV. And if they can cure HIV in cats, there's a higher probability they can cure it in humans as well. And so from this example, when we're looking at what you can actually see here is that this kitten glows in the dark, and that actually means that this kitten has been healed from HIV and is no longer HIV positive. And that is done in a way that they take the virus, and the virus is coupled with a bioluminescent gene, a gene that glows. And so when the virus is destroyed, those genes glow in the different cells of the cat. So this particular kitten has been healed. Here's something very similar in mice. You can see that 3 individuals have been healed or are expressing a virus. You can do it both ways, right? You can attach the bioluminescent gene to the virus if the virus is expressed, or you can attach it to the virus if the virus is destroyed. It will glow, right? You can do it both ways. So here's an example here of 3 individuals that are healed, 3 individuals that are not. And then looking here at this goat, I don't know if you guys can make sense of this, but this actually happens in our own state, right up the road. And there's a group of scientists at Utah State University who are working on this particular problem. And some of you may have figured it out by now, but essentially what they've done is they've taken the genes out of spiders for spider silk production, and they've expressed them in the udders of goats. And so as you milk goats, you get a mixture of both milk and spider's web, and you can purify that spider's web. Here's another example here in flies. So the evolutionary structure of insects is in question, right? How did the different structures form? And what's thought is that all insects were once multi-segmented organisms, and on each one of those segments they had legs. And so the thought is that the head is just one of those ancient segments, and the antennae are just highly modified legs. So what they did is they went in to the antennae and they knocked out a gene that they thought made antennae to see what would happen. And when they knocked that gene out, legs were formed where the antennae should be, kind of confirming this idea that the antennae are just highly modified legs. Pretty cool stuff when you think about it. It's a really cool way to do some genetic engineering. You can also, we talked about some of this bioluminescence as well. Super interesting that it can be done in primates as well, which means it could be done in humans. I don't know how many of you have children in the class, but I have one son who we don't know where he is about 85% of the time. And we've often thought that it'd be nice if he just glowed in the dark, so we could look out into the yard or look around our campsite or wherever we are in the world and find our son. But of course, it's not very ethical, although it sure would make parenting him a little bit easier. Okay, so what is genetic engineering specifically? So it's this alteration of traits by using molecular biological techniques. So it's one or more genes that can be modified within an organism. The genes are generally moved between organisms, and there's a lot of ethics around this, as you can imagine, right? Should we be moving genes between organisms? Is that okay to do? There's a lot that goes on in food and animals and even humans to some extent. One example that we'll talk about is what we call recombinant bovine growth hormone. What is it? In cows, bovine growth hormone increases both body size and milk production. So you can imagine that cows are things that we get milk from and that we eat. So larger body size results in more meat, and more milk results in the ability to sell more milk, right? So in the 80s, Monsanto Company, what they did is they gave bacteria DNA that carries the ability to make bovine growth hormone. And because they moved the gene from the bovine growth hormone gene from cows into bacteria, it's called recombinant bovine growth hormone, or RBGH. And so when we move things between organisms, we call them recombinant. So this recombinant bovine growth hormone is a protein that's coded by a specific gene. So if you transfer the recombinant bovine growth hormone into bacteria, it allows for growth under really ideal conditions. You can get lots and lots and lots of copies of this bovine growth hormone from bacteria. So your bacteria are essentially going to work as factories, and literally overnight you can make billions of copies of this recombinant bovine growth hormone. And then what you can do is bacteria are really special in that in bacteria you have the chromosome that runs the bacteria, but bacteria also have these smaller circular DNAs called plasmids. And these are transferred back and forth between bacteria on their own, but also they provide, because scientists know about them, they provide this really important opportunity to ferry genetic material in and out of a bacterial cell by biological engineering. Again, the bacteria do it normally on their own, but scientists can harness that power to move genes that they want moved between bacteria. So what they do is they use restriction enzymes, which we've covered before, and they cut the enzyme at a very specific way. They can do a blunt end cut right here, or they can do what we call a sticky end cut, where you have these overlaps. And so what you can do, once you know the gene and you have the bacteria and you have your restriction enzyme, you can actually remove the gene from the cow. So you're removing the bovine growth hormone. You can isolate it from the cow. You can cut it out with the restriction enzymes from the cow's DNA. And then you can insert the bovine growth hormone into those plasmids that we talked about here, right? And then after that bovine growth hormone is inserted into the bacteria, it now becomes recombinant bovine growth hormone. Okay, and then the next step is once you've inserted the gene into the plasmid, so here's the recombinant bovine growth hormone in the plasmid, you insert that plasmid within the bacterium. Okay, and then you put that bacterium in like the most perfect environment you could possibly put it in terms of warmth and food and nutrition, and those bacteria will explode. They will just create tons and tons of copies, and essentially creating all the recombinant bovine growth hormone you could ever want. And then what you'll do is you'll take that growth hormone, and you can purify it from the bacteria, and that can be injected into cows to increase milk and size of the cows. About 17% of cows in the U.S. are injected with recombinant bovine growth hormone. Now this is down from about 33%. There were several companies in the U.S., I think namely one of them was Walmart, that said they would no longer carry milk with this recombinant bovine growth hormone in it. So that took the percentage of cows being injected with it down quite a bit. It increases milk volume by about 20%. Now the controversy is whether it's really safe for humans. Should humans be consuming a whole bunch of bovine growth hormone? What are the effects on humans? Because humans and cows, like it or not, are fairly closely related, and especially these genes and how they operate in our bodies and cows' bodies may operate pretty closely in the same way. So that's kind of one of the controversies, right? Is it safe for humans? So the USDA and Monsanto, they argue that the milk with recombinant bovine growth hormone is totally safe for human consumption. A lot of activists disagree. And then there's also things to consider, like the welfare of the cows, right? So Europe and Canada have banned it over the health of the cows. And then we mentioned as well, we eat these cows as well. So what does that have to do with us? I'm not really taking a side here. As a scientist, I can appreciate what's going on, and as a consumer um, of meat and dairy, I can also appreciate what's going on. I would just encourage you to think it through really carefully so that you can kind of understand some of this debate that's been going on. So some other examples of recombinant proteins. So these are the exact same principles apply here. They're made through bacterial generation of these genes, but clotting proteins for hemophiliacs are produced using similar cloning methods to what we just went through. Also insulin for diabetics. Some of you in the class are almost certainly diabetic. Some of your insulin is probably made in a very similar way. Now let's kind of shift gears a little bit and talk about genetically modified foods. So a lot of agricultural products are genetically modified as a result of selective breeding. Now we call this artificial selection. This is essentially like natural selection, which we'll talk about at some point. But instead of it happening naturally out in nature, these are traits that humans decide they like. It doesn't move genes between organisms, but does drastically change the characteristics of a population. So here's an example. This is teosinte, right? This is the ancestor of corn. And then as humans started to eat teosinte and other plants like it, they were able to, over time, create something like this, which resembles more closely what we used to do to corn, and then pushing it all the way to this, which is kind of this big-eared corn. And you actually can't find wild corn that looks like this in nature. It doesn't exist. That is purely a human construct. Okay, so now let's shift to transgenic organisms. So this is the result of when you're moving genes from one organism to the genome of another organism. It's also referred to as genetically modified organisms, or GMOs. You've probably heard that before. Here are some examples here where they actually have taken cold genes from fish that allow fish to tolerate really cold temperatures and put them into tomatoes so that tomatoes could ship better and withstand the cold better. And then pigs and others have definitely been modified in this same way, right? We have the bovine growth hormone as an example. There's similar things that have happened in pigs. So there are reasons why. The question might be, why would you genetically modify foods? Well, there are really good reasons for this. You can increase the shelf life of foods, you can increase the yield of plants, you can even increase the nutritional value. And this may not be something that's super important to us in a developed country that have essentially limitless access to nutrition for the most part. There are a lot of good examples of how this can work in the world. So I don't know how many of you have heard of night blindness. Now I'm going to put up a picture here in a little bit that might be disturbing. I'll try and remember to warn you before I do it. But essentially, night blindness is something that affects a huge part of the world. And it is a lack of vitamin A or beta carotene in diet, and it causes the eyes to become hardened, and obviously you can't see through those as the cornea gets hardened. And so what they do is they can reinforce this rice with vitamin A and beta carotene to fight night blindness. Now I'm going to warn you, I'm going to show you the picture. This is what an extreme case of night blindness looks like. You can see the cornea of the eye has become completely hardened, and this individual is actually blind. Now, this individual is probably too far along to rescue his or her eyesight, but there is the possibility to, if this starts in an individual where there is a hardening of the cornea, if you introduce vitamin A, it can soften the cornea and you can actually rescue the vision of the individual. So here's another GMO that's actually pretty hilarious. This is Alba the bunny. Now Alba was a collaboration between a scientist and an artist, and what they did is they wanted to create a bioluminescent rabbit. And it was thought to be, and it was pitched as This is the most beautiful artwork you could ever create. You can feed it, you can clean up after it, you can cuddle with it, you can spend all this time with this beautiful piece of art that you've created. But it was a huge controversy because on the scientific side of things, they're saying, should we be doing this if there isn't a scientific value associated with the type of research? What should we be doing here? Are we going to start modifying all of our plants, all of our pets? What's going to happen here? And it was really hilarious. This huge, big controversy around it. And then one day, Alba just disappeared, and no one knows what exactly happened to Alba. But I would encourage you to do a Google search on it. It's pretty hilarious, and it all centers around this idea of bioengineering, but even in like a humanities, artistic realm. Here's another one. You can bioengineer chickens to have next to no feathers, at least not the heavy-duty feathers. And these are super ugly, right? But these are really environmentally friendly poultry. One of the biggest byproducts in terms of possible pollution that chicken farms put out are feathers, because they don't break down very well. They're incredibly hard. What do you do with the feathers? And so these are chickens that have been biologically engineered to essentially just have a handful of feathers that can be easily removed, and they are apparently delicious, but they're not so great. So you have to ask yourself, is this happy meat? Is this the future here? There's a lot of good eating meat on that chicken. There's also selective breeding that can be done. So this is a cow that has been selected for a genetic mutation known as double muscle. And these individuals, these individual cows actually have twice the muscle mass of a normal cow. And the question is, should this be what we're going for, right? That we can have twice the amount of meat on a cow. It's not just cows where this happens. You have double muscles, and these double muscle mutations can happen in rats. They can happen in dogs. Here's some pigs and horses and even fish. There are examples in humans. This child here, I think he has his own following, so on social media, but is essentially a double muscle individual. But the mutation for his double muscle is different than the rest of the animal kingdom here. Nonetheless, kid's like 8 years old and looks unbelievably muscular. So there are some effects of GM crops on and the environment. So there are some benefits if you're going to genetically modify foods. So crops that are engineered for pest resistance can clearly resist more pests than those crops that are not. Farmers can spray fewer chemicals because their crops are pest resistant, but there are also some concerns. Are pests going to become resistant to the chemicals that are being produced by the crops? Genetically modified crops may actually lead to an increase in pesticide and herbicide use, because if the crop you're raising is immune to a certain pesticide or herbicide, farmers might spray more over their crop to keep the field as pristine as possible. Genetically modified crops may also transfer genes to wild relatives. Is this a concern? Is this a possibility? These are some of the concerns. Okay, talking about genetically modified humans, there is a great ability. There's a greater and greater ability to do this every year. As a matter of fact, I think a few weeks ago I showed you a headline where it's been okayed in places like the UK, even where it's shown if it can be beneficial to the child and demonstrated to the child that there can be some genetic modification of the embryo. But essentially, when we look at these, one of the most fascinating places for this modification to take place is using stem cells. So stem cells are really just undifferentiated cells. They're embryonic, which means they can grow into many different types of cells and tissue. And they're more of a repair system in adults. If you have adult stem cells, it's more of a repair system, whereas embryonic stem cells there's more possibilities. So stem cells may be able to treat degenerative diseases like Alzheimer's, Parkinson's, MS, liver, lung, and heart disease could be part of this. You can actually grow specific tissues to treat burns, heart attack damage, or replace cartilage and joints. It's really controversial, but the controversy may go away over time, and it appears that it has to some degree. There's also the Human Genome Project. So this was an international effort. It took over 13 years, multi-billion-dollar effort. They sequenced the entire human genome, which is about 20 to 25,000 genes, among 3 billion nucleotides, so among 3 billion A's, C's, T's, and G's. And so for comparison, genomes of other model organisms like E. coli, yeast, and fruit flies were also mapped at the same time. So this cost billions of dollars. And we've had such crazy, awesome advances in technology recently that we can now sequence the entire human genome in just a few hours for about $1,000. And this is absolutely enormous. Technological advance for medicine and other things. And I want you to think for a little bit, if you could have the knowledge in your genome, how would this affect you directly, both good and bad? So go ahead and hit the pause button. Think about that for a minute. Okay, we're going to keep moving. Gene therapy is another interesting way to help humans and potentially things humans depend on, like crops and animals. But in focusing specifically on humans, it's the replacement of a defective gene with a functional gene, and there's two approaches. There's this germline gene therapy and somatic cell gene therapy. But you guys don't know this, but you're already experts on this. For any of you guys who are into like the Avengers side of things, before Tom Holland came on as the current Spider-Man, you had this Spider-Man. And there was this really, from a scientific standpoint at least, a really fascinating movie where you had two individuals within the movie, Spider-Man himself, and then I forget his name, but you have the doctor in there as well, right? And he's trying to do limb generation. And what happens in this movie kind of shows the two different types of germline therapy and the somatic cell gene therapy. So let me just give you a quick breakdown, and then you can kind of decide which one's which. So here, Spider-Man gets bit. The spider injects essentially its genes into it. Those genes go in. They occupy all the cells within Spider-Man. Spider-Man's powers are permanent. And then you have the doctor here who injects himself repeatedly with a set of genes that allow him to regrow his arm, but there's also all sorts of byproducts. He essentially turns into a crazy angry reptile for as long as the injections last. So gene line therapy is what we would call essentially an embryonic treatment. So the embryo, so you go in and you modify the embryo, or you find a way to modify every single cell. And those cells are all supplied with a functional version of the defective gene. The embryo and or cells that are produced by cell division have a functional version of the gene forever and ever and ever. It never changes. Whereas somatic cell therapy is really just a fix or a replace of the defective protein only in specific cells. So all somatic cells, they really have a limited lifespan. The therapy is not permanent, and it requires several treatments per year. And to give you an example, there's one, this severe combined immunodeficiency disorder. It can be treated by somatic cell gene therapy. And essentially what they do here is they remove the immune cells from the patient. Okay, they use a virus to insert a functional immune allele in the immune cells, and then they re-inject that into the child. And if you can tell, she looks really happy about this. Okay, so they inject it into the cells, and then her immune system functions as long as those cells survive. So kind of with this idea, which type of gene therapy does each most closely resemble? What kind of gene therapy did Spider-Man get, and what kind did the good doctor get here? Okay, hopefully you can figure that out. Some other examples of that are potentially on the horizon for humans is cloning. So turns out humans and other organisms as well, but in humans there has been natural cloning for a very long time. And this is in the form of identical twins. So if you have an identical twin, it's really just a clone, and that's where you have sperm and egg that come together, and then the embryo splits, and they're going to share a placenta, and they're going to be exactly similar. I'm really lucky. I actually have identical twins at home. It's really fun to watch all the similarities there. And this is opposed to fraternal dizygotic twins, where there's really no cloning going on here. It's just two separate sperm and egg situations. They do not share a placenta. So cloning has already been done with dogs and cattle and goats and mice, cats, pigs, sheep, all sorts of stuff. Cloning is achieved through the process of nuclear transfer. So that's different than what's happening naturally in humans, and we can kind of go over it here. So this is how you do nuclear transfer. So you take a black-faced sheep, okay, and you take an egg from that black-faced sheep, but you remove the nucleus. Now remember back to our organelles, right? You're taking out essentially all the DNA information, and then you also take you just take a random cell from a white-faced sheep, and you combine the donor egg cell that has no genetic material with the cell from the white-faced sheep, and you put them together. So essentially what you end up with is an egg fused with the genetic material of a white-faced sheep with the egg from a black-faced sheep. So the embryo begins to develop. You put that embryo inside of a black-faced sheep. If the black-faced sheep gives birth to a white-faced lamb, then you know that that is a successful clone. Now, if that doesn't quite make sense to you, work it through a little bit. It totally will, but here's the experiment, right? Here's the mother of Dolly. This is Dolly, right here, Dolly the sheep. So Dolly was cloned from a mammary gland cell of a white-faced mother. The lamb was named Dolly after Dolly Parton. I'll let you put that together. It lived from 1996 to 2003, so about 7 years. It was euthanized because it had severe lung disease and arthritis. So sheep usually live 10 to 12 years. Dolly lived about half that time. Some believe that she showed increased aging due to cloning. So Dolly was born after 277 cloning attempts. There were 3 lambs born, and only Dolly lived. So kind of something to think about, right? I mean, the cloning sounds like, a way forward for a lot of interesting aspects of life. Do we want to clone? Is cloning ethical if there are these types of issues? Is it ethical first and foremost? And if you say yes, we should be able to clone, is it still ethical if you know that there's going to be some sort of health disadvantages to whatever's being cloned? Okay, so here's some of the controversy around genetic engineering. So you have the genetically modified crops. They may make farms more productive, may taste better, longer shelf life, contain more nutrients. They can cure disease and save lives. But there's also these ideas of encouraging agribusiness, which may close down small farms. Animals and crops from genetically modification may cause health problems in consumers. Unexpected adverse effects on the environment. So there could be a lack of genetic diversity in genetically modified crops as well, which could lead to the destruction of food supply. And then kind of this present research might lead to unethical genetic modifications of humans, right? Because a lot of what we're doing in our close relatives, mice, cats, mice and cats and rabbits and even primates, is certainly something that can be done in humans at the same time. So it's all something to think about. Really, these are things that are going to face you in your lifetime, and there are definitely going to be some decisions that are going to have to be made. So that's it for the lecture today. Hopefully this was enjoyable to you. Feel free to look around on the internet about genetic modification. Be really careful and be sure that you're looking at both sides, as you do all the time, both the good and the bad, so that you can fully appreciate everyone's opinions and kind of sort and figure out where you stand. And I hope you're having a great day and that your semester's going well.